Bien, chers collègues, nous allons passer à la lecture suivante qui nous sera délivrée par le professeur Décio Essidric, qui est professeur titulaire et directeur du Centre pour la recherche du diabète à la faculté de médecine de l'ULB. Il a publié plus de 300 articles et critiques dans des revues internationales prestigieuses. Il a reçu de nombreux prix nationaux, internationaux, y compris celui de la Fondation du diabète juvénile. Il a reçu énormément d'autres prix pour ses contributions exceptionnelles dans la recherche sur les îlots de la rivière Hans, prix qui lui a été remis à Berlin en 2012. Et il a encore eu un prix remarquable en 2013 pour ses recherches sur le diabète. Il est répertorié par les indicateurs essentiels Science, ici parmi les 1% les plus cités scientifiques en médecine clinique et biologie et biochimie. Et je pense qu'il va nous faire un exposé certainement passionnant, pas tout à fait, mais peut-être lié quand même à l'obésité, puisqu'il va nous parler du diabète et plus particulièrement de bêta-cell apoptose in type 1 diabète. Monsieur Essidric, je vous donne la parole.
Merci beaucoup, c'est un plaisir pour moi d'être ici, c'est aussi un grand honneur. Et je pense que ça sera... Ça va? Je pense que peut-être sera plus simple que je parle dans un anglais modéré ou bien, et pas dans mon mauvais français. Donc, si vous me permettez, je change maintenant d'anglais en français, mais après, si vous voudrez poser une question en français, ça sera bien aussi. So, what I would like to discuss today is some new findings that we have, mostly connected to the question on why in diabetes type 1 we have a specific destruction of the pancreatic beta cells. And the data that I'm going to show was mostly generated in our own center. It's a collaboration with several colleagues that work with us, particularly Anne of the Beck. And I list below all the funding that have supported our work, with emphasis on Bio, who started to support us last year. And we also collaborate with colleagues with several other centers, and the genetical part is particularly done together with Cécile Juliet and Fleming Possio, and some of the work is also done together with Hotamis Legal in Boston. So when we speak about type 1 diabetes mellitus, this is an autoimmune disease where something goes wrong. We really don't know why, but the immune system starts to recognize pancreatic beta cells as a foreign cell to be destroyed. And when this phenomenon starts, the islets of Langerhans, they will be invaded by macrophages and T cells, and these cells will induce pancreatic beta cells by the pancreatic beta cell death by the apoptotic pathway. And this is done both by cell-to-cell contacts and by release of soluble mediators, particularly the cytokines, interleukin-1 beta, tumor necrosis factor alpha, and interferon gamma. And this is a highly prevalent disease. So in Europe, in the population, between 0.1 to 0.9% will have type 1. And what is particularly concerning is that the incidence of this disease is increasing. So in the last 25 years, it has doubled in Europe, and there is no indication that this trend is changing. So there is a major public health problems here that we need to try to face. When we look at the pancreas of patients with type 1 diabetes, and this figure was done together with Isabel Salmon, who is here, one thing that is really that calls our attention, for instance, in the top figure you have a control individual, in the medium, in the center the islets are stained for insulin, in the left to glucagon, And you can see that in 3 patients with long-standing type 1, nearly all pancreatic beta cells disappeared while the alpha cells remained. So the question is, let's say these cells, they are closely neighbor cells. The immune system is coming in. Why on earth they are killing only pancreatic beta cells while the alpha cells remain? So we have been trying to answer this question, and this is the main issue that I would like to discuss with you today. So over the years, we have developed an hypothesis that I think today is more or less accepted, that the destruction of the pancreatic beta cells in type 1 is not a one-way road. Let's say it's not a situation where the pancreatic beta cells are just sitting there as lame ducks and they are attacked by the immune system. There is more and more data to suggest that there is a dialogue between the invading immune system and the pancreatic beta cells. This dialogue, how this dialogue starts is not clear. It may be, as I am going to propose today, that there is a viral infection. Just a technical detail, the pointer doesn't work. Is there any alternative? Oh, no, okay. Well, perhaps since I have the... Thank you very much. That's the advantage of having a very well-prepared colleague giving a lecture. So, why this dialogue starts remains to be clarified, but one possibility is that there will be a viral infection. And indeed, as I am going to discuss, pancreatic beta cells have receptors, particularly for enterovirus, and they have several mechanisms to recognize this viral infection and trigger an adequate response. The other possibility is that a non-specific inflammation will lead to local production of pro-inflammatory cytokines, leading to activation of cytokine receptor signaling. This will lead to activation of several transcription factors. Perhaps the most important in this context are STAT1 and nuclear factor kappa B. This is regulated, as I am going to show, by some candidate genes for the disease detected at the beta cell level. The next step is what we call the effector phase, where there will be an upregulation of MHC class I, endoplasmic reticulum stress, changes in alternative splicing, which together with pro-apoptotic signaling will lead to death of some of the pancreatic beta cells. Beta cell death, together with endoplasmic reticulum stress and changes in splicing, may lead to presentation of modified antigens in the context of local inflammation. And in general, when we have apoptotic cell death, this is supposed to calm down the immune system. However, under these conditions of a strong local inflammation, these apoptotic beta cells will function as additional danger signals, making this process go on and on. So when we proposed some years ago this concept of a dialogue between beta cells and the immune system, one of the critiques that we usually got was the idea that when we look at the candidate genes for type 1, and today, let's say the genetical inheritance of type 1, this is a disease with a strong genetical inheritance. More than 80% of this genetical inheritance is explained by around 55 genes, where the age LA class 1 are by far the most important. So the prevailing view until a few years ago was that nearly all candidate genes for type 1 would act at the immune system level. So to test if this was correct, we did a simple experiment. We have done RNA sequencing of human islets of Langerhans under different pro-inflammatory conditions. So we just asked how many of these candidate genes were present in human islets of Langerhans. So it turned out that more than 80% of the type 1 candidate genes are present in islets of Langerhans, and at least 50% of these candidate genes modify expression and function during inflammation. As we start to get this type of knowledge, it is possible to make pathway analysis to try to understand what are these candidate genes doing at the pancreatic beta cells. And it's very interesting. All that you see in red are candidate genes. Basically, the candidate genes acting at the beta cell level, they are involved in innate immune response. So you have candidate genes like MDA5, which is involved in the recognition of a viral infection. You have several genes in the pathway leading to type 1 interferon signaling. You have some genes, like for instance PTP-N2, that regulates the dialogue between beta cells and the immune system. You have candidate genes that control the cell death pattern. And you have only 2 candidate genes that control beta cell metabolism and function. It's interesting that this GLIS3 that you see here is the only candidate gene in common between type 1 and type 2. So there has been some suggestions that we should pull together these 2 syndromes. I think this is a basic mistake. I think these 2 syndromes have basically different pathologies and shouldn't be grouped together. So one thing that puzzled us when we look at all these candidate genes and all these complex functions is why on earth, from an evolutionary point of view, will pancreatic beta cells express all these pathways? I mean, to do all this to commit suicide doesn't make sense. So why do they have this? And I was thinking about it when there was a very interesting review in Science where they outlined something which is called cell autonomous immunity. So this is a very ancient response that even, for instance, bacteria have, and this is what allows an infected bacteria to deal with a viral infection. So this continues, let's say, up to mammalians, and when you look at the genes involved in this pattern, you can see that you have genes in the cell autonomous immunity that will counteract all the steps of the viral infection, from invasion, proliferation, and so on. So what we did was to cross this list of genes against genes induced by inflammation in human islets of Langerhans. And indeed, what we found is that when human islets of Langerhans are exposed to a pro-inflammatory response, they trigger several genes involved in this cell autonomous immunity. And one thing that is interesting is that beta cells express several genes like toll-like receptor 3, MDA5, and RIG1 that are involved in the recognition and response to virus. However, they do not express genes involved in antibacterial responses. For instance, they have negligible levels of toll-like receptor 4, suggesting that most probably, from an evolutionary point of view, they may sometimes need to deal with virus, but very seldom with a bacterial infection. So this is an interesting hypothesis. How can we test it? So the way we did was to compare RNA sequencing of human islets of Langerhans exposed either to pro-inflammatory cytokines or to a viral infection. In this case, it's Coxsackie virus B5, which is one of the potential diabetogenic viruses. So this is the baseline. So as you can see, this red and blue line, as they diverge from the baseline, this suggests that there is a correlation. So for instance, if you look at the 20% top genes in viral infected and cytokine treated, there is a more than 50% divergence against base, and there is a more than 50% similarity between these 2 groups of genes. Of course, the next question is whether this is specific or just a non-specific stress response. So together with Miriam Knopp, we treated human islets of Langerhans with we induced a metabolic stress with palmitate, and in this case, palmitate compared against virus, there is no correlation whatsoever. So what we are observing when we deal with virus and with cytokines is not a non-specific stress response. It's a specific genetic signature of a cell dealing with stress. So the next question that one gets when we are finding this type of data is how specific is this for the beta cells? I mean, it could well be that all cells that we have, when faced with a viral infection, they are going to mount a similar response. If this is correct, this would decrease very much the interest of these findings. So we were pondering this issue when I found a very interesting paper in Nature Medicine. And these colleagues, they were studying a viral encephalitis called Nile virus fever. And this encephalitis, it's a mortal disease where there is a preferential damage to the neurons in the cortex while cerebellum are protected. So the cerebellum cells do not die. So what they did in this case was to do an array analysis where they compared cells from the cortex against cells from the cerebellum. And basically what they found is that the cerebellum has a more robust antiviral response. And the key element here is a transcription factor called STAT1. As you will remember, STAT1 was one of the key factors regulated by candidate genes for type 1. So we looked at it and we started to think, well, could it be that a similar phenomenon is present in the islets of Langerhans and explain why beta cells but not alpha cells are killed? So to test this hypothesis, we decided to develop a method to FACS purify pancreatic beta. This was already available, but the big challenge was to purify pancreatic alpha cells in a way that they remain viable in culture. This turned out to be a little bit of a headache, but after 1 year, a very talented postdoc in the lab, Laura Marrocchi, succeeded. And now we can separate alpha and beta cells, and they are more than 90% pure, and they remain viable for more than 2 weeks in culture. So we had in hands a good tool to test this hypothesis. So the first thing that we did was to expose pancreatic beta cells and pancreatic alpha cells to 2 pro-inflammatory cytokines that play a role in type 1, interleukin-1 beta plus interferon gamma. To our complete surprise, there was no difference. So up to now, the knowledge in the literature was that alpha cells are more resistant to cytokines. I think this comes because the purification of alpha cells didn't work very, very well. So it turned out to be that susceptibility to inflammation is identical between alpha and beta cells. So the next step was to see how they respond to a viral infection. And there we got another surprise. So when we infected beta cells and alpha cells with Coxsackie virus B5 at a multiplicity of infection of 5, after 36 hours, you have deaths of 40% of the beta cells, and in the alpha cells there is no cell death at all. So we did a dose response, and in alpha cells you can go to a multiplicity of infection of 100, and still they are less killed than at a multiplicity of infection of 5 in pancreatic beta cells. So alpha cells are more than 20-fold more resistant to this viral infection than beta cells. We confirmed this with another diabetogenic strain, CVB4, and now with another one, CVB1. So this seems to be a broad biological phenomenon. So the first thing that we thought was, okay, for the Coxsackie virus to go into the cell, they need particular receptors. It's CAR and DAF. So it would be conceivable that alpha cells just do not have this receptor. And this turned out to be wrong. When we measured expression of these 2 receptors, they were even more expressed in alpha than in beta cells. Of course, what one of the referees rightly so complained is that the fact that the receptor is there doesn't mean that it works. So we needed to prove that the virus were indeed entering beta cells and alpha cells. So how to do it? What we did was to use an adenoviral vector encoding green fluorescence protein. Why to use an adenoviral vector? Because it entered the cells using the same receptor as the Coxsackie virus. So when we did this in beta and alpha cells, we saw that the number of positive cells was identical, meaning that the virus were indeed entering in alpha and in beta cell. However, when we looked at the intensity of the fluorescence, it was much higher in beta cells than in alpha cells. So what's the message here? The message is that the virus entered in both cells, and then the alpha cell trigger a vigorous antiviral response and block translation and block proliferation. How to look at it further? So the first thing that we did, we looked at our global gene analysis, and we selected the genes that were found in the cerebellum to be involved in the antiviral defense. And we put, this is log scale, when we compare alpha against beta cells, nearly all of these antiviral genes were higher in alpha than in beta cells. We went a step further, we just put together, let's say, the genes that are overexpressed in the cerebellum neurons against cortex, and compared them with the genes that are overexpressed in alpha cells as compared to beta cells. There is a significant correlation, but the most important is here. You see that the vast majority of the genes that are higher in alpha cells, they are also present in the cerebellum, and they are higher as compared to the cortex. So this is all very nice, but up to now these are correlational findings. So how could we prove that there was causation here? So the first thing that we did was a time course response. In blue, you have pancreatic beta cells. In red, alpha cells. VP1 is the viral proteins, the Coxsackie virus capsule protein. So by measuring VP1, we can follow the proliferation of the virus in the cell. You see that in the beta cells, things go progressively wrong. So the virus proliferate, and at this stage, the cells are going to start dying. While in the alpha cell, you have an early proliferation, and then the virus go down. Why alpha cells are able to counterattack this viral infection? Because as in the cerebellum, they have a much higher expression of STAT1, which is basally higher than beta cells, and then it goes up even further. And probably due to this higher expression of STAT1, they have a higher expression of MX1, which is a classical antiviral protein, and of several other antiviral proteins. To test if this was indeed functionally relevant, what we did next was to knock down STAT1 in the alpha cells and see if under these conditions they start to behave as the beta cells. And this turned out to be correct, so we could have a very good knockdown of STAT1. And when you knock down STAT1, you see that there is a decrease in antiviral protein, which allows the virus to proliferate. So if you knock down STAT1, the alpha cells start to suffer, more or less like the beta cell. Well, this was all very nice, but up to now it was mostly in rats. So how can you look at it in human beta cells? So the first thing that we did was to consider if we couldn't look at samples from patients with type 1 diabetes. The problem is that when the disease starts, the autoimmunity has been going on in most of the cases for years. So what we are suggesting here is not that the virus entered beta cells and killed them. The suggestion is that the beta cells are not able to remove the virus. This evolved to a chronic non-cytolytic infection, which contributes to trigger autoimmunity. So if we are going to look in samples from type 1 patients, this is a chronic situation, you will have, it's still observed that the viral particles are present in beta cells, and they are not anymore present in the alpha cells. Why this? Either because they never entered alpha cells, or because they entered and they were kicked out. So one way to test this, together with some colleagues in the UK, Noel Morgan, we got access to samples of young children who unfortunately died in the context of a very severe Coxsackie virus infection. So this was an overwhelming and acute infection. And when we look at the samples, you look at the VP1, which is the viral protein, and then you look at a glucagon, you can see that there is indeed viral infection in the human alpha cells, at least under these extreme conditions. And we reproduce this under in vitro conditions. So we receive periodically human islets of Langerhans. They were dispersed and infected with CVB Coxsackie virus B5 for 8 hours. And you can see, for instance, if you look at this cell here, this cell is glucagon positive and is positive for the viral protein. So there is a double staining. So at least under this extreme viral infection in vivo and under these in vitro conditions, we can observe viral infections in alpha cells. And now last week we just got data with the adenovirus green fluorescence protein, also confirming that the virus can enter in the alpha cell. So which are the take-home messages? I think studies in human islets combining bioinformatics and molecular biology suggest that more than 80% of the candidate genes for type 1 diabetes are expressed in human islets of Langerhans. Pathway analysis of these candidate genes identified a central role for interferon-regulated pathways and genes such as MDA5, PTPN2, and TYK2. We suggest that type 1 diabetes candidate genes play a role in regulating beta cell defense or cell autonomous immune response against infections, particularly viral infections, or other danger signals. So I don't think that this is specific only for the viral infections. And I think perhaps the most intriguing concept is that pancreatic alpha and beta cells have different cell autonomous immune responses, which may explain why beta but not alpha cells are destroyed in type 1. Thank you very much for your attention.
Thank you very much, professeur Eziric. Maintenant, je donne la parole à qui la demandera. Monsieur Goldman.
Very nice talk. As you mentioned, type 1 was, or is still primarily considered as an autoimmune disease, right? Mm-hmm. What you show here is what could be the trigger of autoimmunity. Now, as you know, most therapeutic intervention, beside insulin replacement, aims at restoring tolerance, right, to beta cells. And so, would you suggest that we should now look for other type of therapies targeting the very early steps related to indeed inflammation?
Yeah, that's a very good question. So there are trials starting for vaccination against Coxsackie virus. So this is it's something that's being done in Finland, and the idea would be to try to prevent disease by preventing the virus from entering beta cells. One thing that we need to be careful here, and this is a very interesting story, many of the candidate genes that we find acting in the beta cell, they actually lead to an extra vigorous response. So it seems that the beta cell needs to have a very tight balance because the cells proliferate very, very little. They are like neurons. So for instance, if you have a gut viral infection, you get rid of the virus by having most of the cells infected dying. In the beta cells, they cannot do it, because if 80 or 90% of the beta cells die, you cannot recover them. So they need to find a way to live together with this viral infection. So it seems to be that if you have an excessive or an inadequate response to get the virus out, this increases the chance in individuals with a particular genetic background to develop autoimmunity. So if we come back to your question, vaccination should be done to prevent that the virus enter. We wouldn't like to vaccine to boost the immune response once the virus is in, because this perhaps could have very bad consequences. So this is something that will be tried. The other challenge is which virus to target. So there are by now 3 or 4 candidates. And in most cases, when disease started, the viruses are already gone. So it's a little bit difficult to know which of them to target.
Oui, madame.
Thank you very much for the very nice presentation and very nice data, really. So what I would like to ask you, so there is now in the northern countries an increase of the type 1 diabetes onset in the children. Mm-hmm. And we see also the increase in our countries later on in life. So do you think that the age may play a role in the deference towards the viruses, or if the contamination with viruses could explain these two types of
Yeah, that's a very good question also. So the prevalence, the incidence, as I say, is increasing, and now the fastest increasing is in the former Eastern European countries. And there are now big prospective studies, and it seems that the autoimmunity starts very early, in the first 6 to 12 months of life, much earlier than what we imagined before. We don't know what is the cause. I think there are 3 things going on. One is the genetic background, but this just explains part of the story. For instance, identical twins, if one has type 1, the other has a 30 to 60% chance of developing disease. So there is a genetic component. There is perhaps a role for virus infection, but the third complicating issue, especially in the Nordic countries, is the so-called hygienic hypothesis. So the idea is that in these countries, and also here, the very good hygiene early in life hampers the education of the immune system. So when the immune system is faced, for instance, with a long-term intra-beta cell viral infection, they may react in an excessive way. So we just wrote a review together with Anne of the back. So what we are proposing is that people were trying to find the role of virus in type 1, looking at the Koch postulates. But I don't think that this will work out here. I think here we are dealing with genetic background, hygiene or education of the immune system, and virus. So we need to take these 3 things into consideration. And I think one study that will provide the answer to this question is a big epidemiological Finnish study. This is an interesting story that mixes politics, geography, and public health. So in Finland you have an area called Karelia, and in the Second World War, Finland lost half of Karelia to the then Soviet Union. So this population is genetically identical, but the prevalence of type 1 is 4 to 6-fold higher in the Finnish Karelia than in the Russian Karelia. So now there is a prospective study where they are looking at the bacteriome, they are looking at the immune response, and they are looking at candidate genes, and of course at viral infection. And it's funny because, well, it's not funny, it's tragic. It seems that the baseline autoimmunity is similar. So the HLA class 1 is similar, even the prevalence of autoantibodies is similar. But while in the Russian side they have autoantibodies that they don't evolve to disease, in the Finnish side they do, which may perhaps due to the hygiene hypothesis. But this is an ongoing study. We should know in more or less 3 to 4 years.
Merci. Gustave?
A short question. You mentioned a comparison between cerebellum and Serracolex as compared to alpha and beta cells. It turns that cerebellum is phylogenetically much more older than the cerebral cortex. I mean, the cerebellum of a navigator is no different at the microscope from the cerebellum of a human. So is there a difference in the phylogeny of alpha and beta cells?
That's an interesting question. No, actually, they have a similar. Let me just think a little bit. For instance, in fish, they have something called Bruker body, which is a big islet of Langerhans, and this is consists mostly of beta cells. In birds, for instance, in ducks, alpha cells are more important than beta cells. So, for instance, if you do pancreatectomy in a duck, they die of hypoglycemia. Funny enough, it's the opposite. But I think they have the same genetical background, and now there are a lot of studies showing that it's possible in mammalians to transform, for instance, beta cell in alpha cells and the other way around. So, to my knowledge, there is no data to suggest that from an evolutionary point of view, alpha cells come early. But I will look at it. That's an interesting possibility.
Thank you. Any other question? Yeah. So thank you very much for your very